Good afternoon, my name is Mesa.
I'm Rajschmir.
And I'm Gucciardo. And we are part of the learning community here at Miami Dade Wolfson campus. We are currently enrolled in General Chemistry 2, Principles of Biology 1, with Professor David Finneran and Professor Juan Morata.
This afternoon we will be presenting our final research project where we tested the inhibitive antimicrobial properties of Florida orange blossom honey, raw agave, and blackstrap molasses against 4 different strands of bacteria: Bacillus cereus, Escherichia coli, Pseudomonas aeruginosa, and Staphylococcus aureus. For our project, we used our Florida orange blossom honey, raw and unfiltered, sourced directly from West Palm Beach, Florida. When a honeybee collects nectar from a flower, nectar is primarily sucrose with a water content of 70%. They take it back to the hive in their stomach. In their stomach, enzymes break up the disaccharide sucrose into monosaccharides, glucose, and fructose, or dextrose and levulose, as they are also known as. Once the honeybee goes back to the hive, the home bees, as the beekeepers call them, will take in the regurgitated nectar that honeybee brought back, and they will continue the process of breaking down more sucrose into the monosaccharides, glucose, and fructose. Once that process has been complete and the sucrose has been broken down sufficiently to be put into the hive, the home bees will place the honey into the hive and start fanning the honey so that the water evaporates out of the honey, lowering that 70% concentration from the nectar to a low 17% water concentration in most honeys. Honey also contains several carboxylic acids, which make it highly acidic at a pH of 3 to 4. Carboxylic acids like formic acid, in this particular honey citric acid, since it is sourced from citrus, and gluconic acid, which is the predominant acid found in honey, these all attribute to the very low acidic pH. Hydrogen peroxide is a product of the oxidation of glucose into gluconic acid. Hydrogen peroxide is also known to attribute many antimicrobial properties to honey. Of 17 flavanones identified in orange blossom honey, the highest concentration is hesperidin. Hesperidin also has been proven to have anti-inflammatory and antimicrobial properties, and all of this makes us believe that honey will have the highest antimicrobial properties and the highest inhibition of the 4 strains of bacteria that we will be testing.
The agave that we used is Agave tequilana. It is raw agave native to Mexico. Agave tequilana is processed by chopping off the stem of the plant and bringing it to a processing plant where it can be treated with high temperature and high pressure. Agave tequilana contains 70% fructose and 26% glucose. Its pH fluctuates around 3.8 and its moisture content is 22.5%.
Blackstrap unsulfured molasses is made by the pressing of sugarcane. The juice extracted from the sugarcane is then put to a boil to promote crystallization. The first boil is known as cane syrup, and the third boil is known as blackstrap molasses. The properties of blackstrap molasses are calcium, selenium, copper, manganese, magnesium, and iron.
Originally, we wanted to conduct this experiment using garlic and ginger as well as honey. We are aware that garlic and ginger both have antimicrobial properties, and we wanted to compare the 3 to see which one had the most antimicrobial properties. Unfortunately, we encountered several difficulties when preparing our garlic and ginger to make our nutrient agar concentrations. In order to get the ginger and garlic concentrations into the agar, we decided that the best way to do it, and the most accurate way to do it, would be to dehydrate our ginger and garlic and grind it into a fine powder so that it could be the same sort of material that the dehydrated nutrient broth and nutrient agar were. In order to prepare our ginger and garlic for our concentrated nutrient agars, we took the ginger rhizomes and grated them and laid them out in a thin layer over aluminum foil, placed them in the oven at the lowest temperature that the oven would allow, which was 170 degrees Fahrenheit, and continued the process of dehydration by allowing it to sit in the oven at that low temperature for over 8 hours with the oven door open in order to circulate airflow. After this process was complete, we took our ginger and we used a spice grinder to grind the ginger into a fine powder. We then sifted the ginger powder in order to remove all the fibers that were insoluble in the nutrient agar. The process for the garlic was very similar to the ginger. I peeled and took apart the cloves of garlic, and then I put them into a food processor which ground the garlic into tiny little bits. Then I laid out the garlic on a thin layer of aluminum and repeated the same dehydration process that was used for the ginger. I also ground the garlic into a fine spice with the spice grinder. However, when it was time to add our ginger and our garlic to our nutrient agar, we found that the 75 grams of ginger needed for a 30% concentration was too much, and it was oversaturating our nutrient agar solution. We hadn't even added about half of the 75 grams when we realized that it was not going to work and we had to scrap our idea. Our next step was to find 2 more substances that were just as soluble in the nutrient agar as the honey was, because the honey was so successful in mixing into our concentrations. In the microbiology lab, we prepared our nutrient agar. The microbiology lab houses sterile conditions to work with nutrient agar and bacteria. First, we tried to make our 15 and 30% concentrations just to see if they would work. For 15% concentration, the amount of honey, agave, and molasses that needed to be used would be 37.5 grams. For the 30% concentration, 75 grams would have to be used in proportion to the nutrient agar and the nutrient broth and water. First, we took distilled water and measured it in a graduated cylinder and measured out 250 milliliters of that distilled water. We poured that into a 500 milliliter flask, Erlenmeyer flask, and we slid a stir bar, a magnetic rod, into it. We covered it with a layer of aluminum foil. We placed a little tape, autoclave tape, right on top to signify that it had not gone into the autoclave yet. And we repeated the process several times according to how many different concentrations we had. After we placed the 250 milliliters of distilled water into each Erlenmeyer flask, we went ahead and measured out 3.25 grams of nutrient broth, as well as 3.75 grams of nutrient agar. Both of these are dehydrated. We tared our scale to 0, measured out the weighing boat, tared it to 0 again, and then slowly began to add into the weighing boat 3.25 grams of nutrient broth. First, we added the nutrient broth into each Erlenmeyer flask. During this process, my lab partners were preparing our hot plates. We laid out several hot plates. at 90 degrees Celsius on the bench. In the meantime, I weighed out 3.75 grams of nutrient agar and added that to the nutrient broth and the water. We placed our Erlenmeyer flasks on the hot plates and began to stir them slowly as we measured out our honey, agave, and molasses. For 5% concentration, we measured out 12.5 grams of each, honey, agave, and molasses. For our 10% concentration, 25 grams were used. For our 15% concentration, 37.5 grams were used. 20% concentration, 50 grams. 25% concentration, 62.5 grams. And for 30% concentration, a total of 75 grams were used. As our Erlenmeyer flasks sat on the hot plates and stirred, we proceeded to add our different amounts of honey, agave, and molasses into the Erlenmeyer flasks. Viscosity is reduced as temperature increases. We placed the beaker onto the hot plate and stirred it with our solution in order to not have to add any more water. And then we poured the leftovers of the beaker into the Erlenmeyer flask to make sure that all of our materials were being used at the appropriate concentration. After letting the solutions stir enough before they boiled, but enough so that they were completely soluble in the water, we went ahead and placed our Erlenmeyer flasks into the autoclave. The autoclave is a machine that completely sterilizes anything you put into it at a very high heat and pressure. This sterilized all of our materials before we poured them into our Petri dishes. In preparation of pouring our nutrient agar solutions into the Petri dishes, we sterilized the bench with disinfectant, lit several Bunsen burners, and left them lit while we opened up our plate bags. The plates are sterile until you open them, and we wanted to maintain the sterility of the environment. We laid out 60 plates onto the bench. We took out our nutrient agar solutions from the autoclave. We used a rubber glove to hold the Erlenmeyer flask by the neck and pour them slowly into the nutrient agar plates. Due to the different concentrations, the volume increased accordingly. So 5% yielded less plates than 15%, and 15 less than 30. We slowly poured enough nutrient agar into our Petri dishes so as to cover the full bottom surface of the Petri dish. We ever so lightly removed the cap, poured it inside, and covered it back up so as to not contaminate. We let them sit and we let them harden for about 20 minutes. After they were hard, we went ahead and flipped over our Petri dishes and we labeled them according to the percent and according to the material being used, as in 5% nutrient agar honey concentrate or 15% agave nutrient agar concentrate. We went further and we drew quadrants onto our Petri dishes and labeled each quadrant accordingly to the bacteria strain that we would be adding to that we would be inoculating in the agar.
During our inoculation stage, we sanitized our workbench, lit our Bunsen burners, and grabbed our inoculation wands. We sanitized our inoculation wands by burning them in the Bunsen burner. We then grabbed our nutrient agar plates with our appropriate concentrations, uncovered them slightly by the Bunsen burner, and inoculated the nutrient agar with the appropriate bacteria in the appropriate quadrant.
Once we observed significant inhibition in the molasses agar of 15 and 30% concentration, we went ahead and we did 4 more concentrations of 5, 10, 20, and 25. After being incubated for 24 hours, we were ready to inoculate. But now we discovered we had mold on our agar. So why is it that our molasses agar plates of 15, 10, 20, and 25% grew mold? Our explanation was that our jar of blackstrap molasses became contaminated between the first batch and the second batch of nutrient agar. In retrospect, as a group, we do remember observing water precipitation in our molasses agar plates. As a group, we concluded that the excess water promoted production of mold on our molasses agar plates.
24 plates of plain nutrient agar were gathered from the microbiology lab. Then, 12 of the plates were quadranted to be labeled 5, 10, 15, 20, while the other half, 25, 50, 75, 100%. Later, 6 plates of all of those concentrations were streaked with the 4 different types of bacteria we inoculated. Later, we made the various solutions by adding, for example, in the 5%, we measured out 9.5 milliliters of distilled water and mixed it with 0.5 milliliters of honey. We later took those solutions and dipped in a sterilized hole punch into them. Then we carefully placed them in their respective agar plates with the bacteria on them. Later, we singled out 4 different plates and added ampicillin in each of those as a control. We took those to the biolab and incubated them for 24 hours at a temperature of 37 degrees. As we can see from the pictures, the bacteria on the plates were not inhibited. This can be due to many reasons. The bacteria were not inhibited because we hypothesized that since all the bacteria were not covered in the nutrient agar, probably it did not have enough, the water quantity did not have enough potential to get sucked out of the bacteria, causing it to still live. As we can see from the halo around the bacteria inside this circle could be the killed bacteria, but not completely killed. Even in the 100% concentration, the bacteria was still growing. The bacteria was still growing since the water did not pull away from the bacteria. Also, there could have been some experimental values that could have gone wrong. In one of the plates, the ampicillin still didn't inhibit the bacteria, but in another one it did. In this method, the standard test for antibiotics, the test that we did on honey did not work.
Overall, honey inhibited all of the bacteria that were experimented on. Whether it was gram-positive or gram-negative, it did not matter because it inhibited all of them. Moisture content played a direct role in the inhibition rates of honey and agave. The concentration of honey is directly proportional to the inhibition or growth of all of the bacteria. Firstly, we will examine different types of bacteria and we will extrapolate the data to see which bacteria inhibited most or least in the various mixtures. Bacillus cereus had a maximum inhibition concentration of 20% in honey and 15% in agave. As we can see from the curve, there is a downward exponential slope that we can clearly see. Next, we have E. coli. The minimum inhibition concentration was 30% in honey and 30% in agave. Honey showed to be the most inhibitive with E. coli. As we can see from the graph, the 15% concentration is an outlier. Some experimental factors could have been attributed to the outlier in this graph. Further experimentation must be taken to determine a new concentration and then later see if that outlier may be intentional or it could have been an experimental error. This would be an accurate method to calculate the results. Next, we had Pseudomonas with the minimum inhibition concentration of 25% in honey and 25% in agave. It is noted that in 15% molasses, no Pseudomonas grew. From the graph, we can clearly see that Pseudomonas grew more in 5% honey than 0% honey. Further experimentation must be taken to determine a new concentration and then later see if that outlier may be intentional or it could have been an experimental error. This would be an accurate method to calculate the results. This fact negates our hypothesis because our group thought that there would be a more inhibition rate in the higher concentrations of honey. This is true to all bacteria except Pseudomonas. This is because Pseudomonas is special in a sense that it thrives in higher concentrations of carbon. That we know for a fact that honey and agave are mostly made up of complex and simple sugars with the main component as carbon. Next, we have Staphylococcus aureus. The minimum inhibition rate for this bacteria is
30%
in honey and
30%
in agave. We can see also in this bacteria, like E. coli, the 15% data is an outlier again. Staph grew the least in all the mediums that we put in. This can be due to many reasons, one being that the optimal pH of staph is 7 to 7.5, which is slightly more basic than the rest of the bacteria, which usually grow at a pH of 6 to 7. Because the nutrient agar is a pH of 6.8, and by adding honey or agave a pH ranging from 3 to 4, we make the concentration more acidic, a non-ideal thriving ground for staph. Overall, we can see that honey inhibited growth. However, with certain types of bacteria, they behave differently. When conducting this experiment in the future, take into account that different types of bacteria react differently with different types of concentrations of sucrose and glucose.
As many may know, honey has medicinal and antimicrobial properties. Honey has been accepted as an agent for the treatment of ulcers, bed sores, and other skin infections that occur after being wounded or burned. Honey's high viscosity can provide a barrier and prevent infection. Honey may promote the repair of damaged stomach lining and intestinal mucosa.